All right, so I'm going to switch gears and talk a little bit about some of the work we've done over the past several years to try and really develop the zebrafish as a platform for cancer modeling. As many of you know, the zebrafish really came into being as a model organism probably in the 60s and 70s and early 80s, primarily because of its capacity for doing unbiased forward genetic screens primarily. And this was coupled with the fact of its really excellent vertebrate conservation of gene function and obviously optically transparent for in vivo imaging. In the past really 5 to 10 years, really what changed in the zebrafish world is our facility for doing very rapid and high throughput transgenesis, small molecule screens, and really both of these really led themselves to development of the model as for cancer biology. So several years ago, we began to think about different ways for developing the zebrafish as a platform for doing cancer biology, and a postdoc in the Zahn laboratory, where I'm a research fellow, began to ask whether she could model melanoma in the zebrafish. And the way she approached this was to use what at the time was the gene that had just been cloned, which is the BRAF V600E mutation, which you heard about this morning. And so this is a wild-type zebrafish here. They're about an inch long. When she overexpressed the human version of BRAF V600E under the control of the melanocyte-specific MITF promoter, these were the animals that resulted, and these are stable transgenics. And what you can see most clearly is that the black stripes, which are normally very organized in the fish, become highly disrupted and really coalesce into these almost hyperpigmented lesions throughout the body of the animal. Now, interestingly, these animals by themselves really never went on to develop melanoma, except in extremely rare cases. But when they were crossed with a p53 loss of function mutant, so across these two animals, these are the animals that resulted. And so I'll refer to these as the BRAF p53 model because they essentially developed these very aggressive invasive melanomas in addition to other pigmentation and stripe abnormalities. And so we began looking at this model in great depth. And so this is work done by Craig Seal, a postdoc in the Zahn laboratory along with me, where he first characterized essentially the onset of tumor development. So these are tumor development curves over the course of a year. And what you can see are really two things from the slide. One is that virtually all animals, so essentially 100% of animals, develop these invasive melanomas by about 12 months of age. There's a latency of about 4 months, and so pretty much every animal between 4 and 12 months develops a tumor. It is dependent on the dose of BRAF, so a single copy of BRAF is here and a double copy, or the other way around. So double or homozygous BRAF develop tumors much more quickly, which is really not too surprising. And so this early work in the zebrafish melanoma model really set us up for thinking about really taking advantage of the zebrafish as a platform for exploring new targets and pathways in particularly melanoma, but more recently other tumors as well. And the reason we thought it was a really excellent platform for doing modifier screens had to do with the fact that it was a very high penetrance model. The tumors develop in very stereotyped locations, and this turns out to be key because we always know where to look, and this makes screening thousands and thousands of animals much more quickly. And so these, along with all the other reasons I talked about, we began to think of it as a fairly ideal platform for two types of modifier screens, and I'm going to talk about two today, and obviously there are others going on. And the first is a genetic modifier screen, and the second is a chemical modifier screen. And I think both of these highlight really the strengths of the system. So the genetic modifier screen was work done by some of the people who are research fellows and postdocs with me in the Zahn lab, Yuriv Huvras and Craig Seal, and I'll talk about this one first, which was a cooperating oncogene approach, and then I'll talk in more depth about a chemical screening approach, which was a small molecule developmental approach done by myself, but I'll talk about each of these in turn. I'll start with the genetic modifier screen. And so essentially, Uriv and Craig were interested in essentially a functional genomics approach in the zebrafish. And really the question that many of us are grappling with, I think every person in this room, is to identify which human oncogenomic alterations have actual biological consequences. In other words, which are the drivers and which are the passengers. And really the goal was to use the BRAF P53 model to really find candidate genes that accelerate or slow down the melanoma in this model. And so they began a collaboration with Levi Garraway's lab at the Broad and at Dana-Farber, and this is work that Levi published a few years ago, where they began to look at amplifications and deletions across a panel of short-term cultures of about 90 melanomas. And obviously these are the different chromosomes here, and there's a variety of commonly well-known amplified genes such as MITF and BRAF and cyclin D1 itself. But there was this region on 1q21 that they chose to focus on because there were no known or obvious oncogenes in this region. It was obviously highly amplified in this set of melanomas. And so this was initially what got them started thinking about how could we query that region. And it turns out that 1q21 is amplified not just in melanoma, but actually in several other tumor types, including breast tumors. And so these are gistic plots showing amplifications in red, deletions in blue across a number of different tumors. And you could see that, for instance, in this panel of breast tumors, many show amplification of genes in the chromosome 1q21 region, and similarly for small cell lung cancer and non-small cell lung cancer. And so when you drill down and look at this chromosomal interval, there were a number of candidate genes, and actually this is only a partial list, turned out to be about 30 candidate genes in this region, which any of these really could have been the region, the genes that were driving the oncogenic changes. And so this was really the gene list that Uriv and Craig started with as candidate genes. So how did they do this? They utilized an overexpression platform that they call Mini Cooper, which stands for Cooperating Oncogene. I didn't make up the name, obviously. And Mini Cooper consists of two different injection plasmids. The first is an MITF mini gene, which consists of the MITF promoter and an open reading frame for MITF. And the second is the Mini Cooper expression vector itself, which has an MITF promoter driving any candidate oncogene that you wish. And the reason that they adopted this model was actually pretty simple. They injected these into an albino zebrafish, which was actually mutated for the MITF gene itself, so it has no melanocytes and never develops melanoma. If you express the MITF mini gene by itself, you get melanocyte rescue on a very clean background. And then if you express the MITF mini gene along in the context of BRAF and P53, you essentially have a very clean background with these very localized tumors. So again, this aids us in screening for animals because they are, they're only an inch, and we're screening in some cases, tens of thousands of animals. So this is a very clever modification of the system that allows us to put in essentially any candidate oncogene. And so they started out by testing some candidates. In this case, they put the first gene as cargo was GFP, and you get a GFP positive tumor as expected. But then they tested a sort of positive control, which was the gene cyclin D1. So these are GFP or control BRAF P53 tumors, and this is the tumor onset over time. And you can see in the presence of concomitant cyclin D1, you get much more rapid tumor onset. And so this was sort of proof of principle that you could pull out cooperating oncogenes using the Mini Cooper system. And obviously, this worked fairly well. So now they went back to their list of candidate genes and essentially began interrogating all of them. And so over the course of this experiment, they essentially generated about 3,000 transgenic animals, which I think is one of the advantages of the fish, is that 3,000 sounds like a lot, but it's actually not, intolerable to generate that. And so what you can see is these are tumor curves, and the GFP curves are kind of in the middle here. And what was fairly obvious from this is that on two separate occasions, really only one gene accelerated tumor onset, and that was the histone methyltransferase SETDB1. And you can see that these animals get tumors much sooner and with much higher penetrance compared to either GFP or all these other genes in the interval. And so this really sort of showed us that they could pull out from a list of genes and really pull out what is the relevant likely driver gene in here. And so they went and looked at a variety of human tissues as well, and they really focused on melanoma. And what's important is that if you look on the bottom here, which is skin, you can see that the majority of skin actually has very low expression of SETDB1. Whereas you go into a nevus or a pigmented mole, they become more intermediate for SETDB1. And obviously, as you get into a aggressive melanoma, virtually all are either high or intermediate level of SETDB1 expression. So SETDB1 clearly correlates with the malignancy of the disease, and which goes along with what we were seeing with the amplification data. So what does SETDB1 do? Well, one of the observations that Craig and Yariv had made is if you look at the effect of overexpressing BRAF alone, what he would often find is that if you look at foci of senescence stained by beta-gal, and this is a scale from a fish, which is sort of like the equivalent of our skin, you can see that BRAF alone often induces these senescent lesions throughout the scale. And if you look down here, if you overexpress in our mini Cooper system with GFP, you see these senescent foci. So it was clear that at a cellular level, what SETDB1 was doing was essentially preventing senescence of these lesions. So the animals went on to develop tumors in part because of suppression of senescence in this lesion. Oops. I think there's a slide missing. But so the question, of course, is how does SETDB1 achieve the cellular phenotype? And I'm not going to go into a lot of details because Craig and Yariv in the Zahn laboratory have spent a lot of time looking at exactly what the targets of the histomethyltransferase are. And I'll say in a short way that what is fairly clear is that the SETDB1 binds to and appears to repress members of the Hox locus, and particularly HOXA1. And so the hypothesis is that by binding to this region and repressing HOXA1 locus, that this is somehow related to the cellular phenotype. But as I said, the details of that are still being worked out, and I'll leave that for now so I could talk about a couple of other things. So I'll switch gears and talk now about our chemical modifier approach, which is really the other major thing we've done with the melanomas. And really early in the course of working with the melanomas, we began to perform expression profiling of the zebrafish melanomas. And what we noticed very early on is that these BRAF P53 melanomas, in addition to being invasive, et cetera, tended to express very high levels of a number of genes associated with the neural crest stem or progenitor cell, and particularly the genes crestin, SOX10, and EDNRB. And in general, they express surprisingly very low levels of the melanocytic markers DCT and tyrosinase. And so this is a zebrafish, much like the one I've showed you previously. This is a wild-type fish sectioned longitudinally, so that's its eye, that's its belly, that's its dorsum. And you can see when you stain it for crestin, this embryonic neural crest stem cell marker, it's essentially completely absent, which is what you would expect. But in the tumors stained purple here, you can see virtually the entire tumor is positive for this early embryonic neural crest marker. And what this really suggested to us is that many melanomas recapitulated early neural crest development or early embryonic development. This turns out to be true in human melanoma as well. So this is a tissue microarray of 70 human melanomas stained again for early neural crest markers like EDNRB and EDN3, where you can see almost 80% express these early neural crest markers. Only about half or a little more than half express MITF, and again, only about 15% express the differentiation marker dopachrome tautomerase. And so when you take the zebrafish and the human together, it really suggested to us that many melanomas, certainly not all, recapitulate early neural crest development and specifically express markers associated with the neural crest stem and progenitor cells, and much less so the differentiation genes. So we decided to take a small molecule approach to this problem to ask, because of this observation, could we use the zebrafish embryo or the zebrafish system to find small molecule in vivo suppressors of the neural crest stem cell program, something that wouldn't easily be done in a cell culture system? And so the way we did the screen was we mated adult fish and generated thousands of embryos. And so in this particular screen, we generated about 25,000 animals. We plated them into individual wells, and then each well got a different chemical. We had about 10 embryos per well. And then we stained them for a variety of early embryonic neural crest markers, as I stated. And these are sort of the purple cells, which each represent an early neural crest stem or progenitor cell in this assay. And so we got a number of interesting chemical hits from this. I'm really going to talk about two today. So this is our control-treated animal, and as I said, this is stained with the marker crestin. MITF itself looks fairly similar, where each of the purple cells are a neural crest progenitor. And you can see in the presence of these two structurally distinct molecules, NSC-210-627 and leflunomide, we see an almost complete aggregation of early neural crest expression. There is a small amount of residual neural crest expression here, and essentially none throughout the trunk of the animal. And you could see the majority of the embryo is fairly intact. This is its eyes, and you can see eyes there, and this is its yolk, and that's its tail. So the embryo is relatively intact. Both of these molecules inhibit an enzyme called dihydroorotate dehydrogenase. DHODH has never previously been implicated in neural crest development until fairly recently, when patients with Miller syndrome, actually a very rare craniofacial abnormality with many features of a neural crestopathy, were found to have inactivating mutations in DHODH. And we really don't know very much about the enzyme itself. So the first question we really asked is, does the small molecule, and the one we pretty much use for everything is the small molecule leflunomide, and we asked, not only does it only affect expression of the marker genes, or does it affect the actual cell types that are derived from the neural crest? Because we weren't interested in just marker genes. And so this is a zebrafish embryo. This is its eye and its yolk at around 2 days of life. Each of those black cells are the actual pigmented cells, or actual pigmented melanocytes. This is in a control and in the presence of our small molecule, and you'll see that these animals are essentially completely albino. This is accompanied by an almost complete loss of an MITF-GFP transgene, where you see GFP normally throughout the animal, whereas in the leflunomide-treated embryos, we see a small residual amount of MITF up here, and another neural crest derivative, myelin basic protein, marked by M-cherry in this case, also completely absent in the presence of our small molecule. So leflunomide appears to suppress not only expression of neural crest marker genes, but also of the derivative cell types as well. So to ask at what level the small molecule was working, we turned to a collaboration with Sean Morrison, then at the University of Michigan, now at UT Southwestern, to ask, does leflunomide affect the ability of neural crest stem cells in a mammalian system to self-renew? Because we saw numerous derivatives affected, and so we sort of assumed that it must be working at the level of the stem cell. And what he did is he isolates neural crest stem cells freshly from a rat sciatic nerve, and then does a replating assay, essentially a self-renewal assay. And you can see in the presence of either low or high doses of leflunomide, you see a very strong suppression of self-renewal of the neural crest stem cell population. And it really does not affect any of the differentiated progeny per se. It really only affects the stem cell itself. And this goes along with some expression profiling we've done, where it shows that DHODH, the enzyme itself, is preferentially expressed in the stem cell population compared to the differentiated population. So while working on trying to understand the mechanism by which leflunomide affected these effects, we noted that leflunomide essentially or strongly phenocopied a particular class of mutations in the zebrafish embryo, all of which were centered around the gene SPT5. And so here I'm showing a GSEA plot where we compare the gene expression profile of embryos treated with leflunomide compared to embryos that have been mutated for the SPT5 gene, an inactivating mutation. And what you can see on both the down and the upregulated plot, we can see an almost complete recapitulation of SPT5 mutation by our drug. And we have other data that I don't have time to show that shows that leflunomide essentially acts as a complete loss of function of the SPT5 transcriptional elongation factor. And so as has been alluded to before, obviously the regulation of activation of POL2 transcription really transitions from a state of POL2 recruitment to the promoter region into the active elongation phase, which counts on many factors, not the least of which is the gene SBT5 itself. And that's really what it's most closely associated with, is the ability for POL2 to actively move into the elongating phase of transcription. And it appeared that our drug leflunomide really specifically interacted with this aspect of SBT5 function, or its elongation function. So to determine whether this was indeed the mechanism by which the drug was working, we collaborated with Rick Young's lab at the Whitehead, who has a lot of expertise in looking at elongation, using a ChIP-seq assay in which they measure RNA POL2 binding across chromatin. And they do the following calculation where they can measure the amount of POL2, RNA POL2, bound in the promoter region versus the amount of POL2 bound in the gene body. And this gives us a very convenient numeric aspect of elongation called the traveling ratio. Where you can see is for genes that have lots of POL2 in the promoter but almost none in the body, the traveling ratio will be very high. And that as it leaves the promoter region and moves into the body, this ratio will go down. So the traveling ratio essentially acts as a readout of transcriptional elongation at essentially genome-wide. And so we utilize this assay in human melanoma cells. And before I talk about what leflunomide did to that, I think one of the really important things that we and others are recognizing about transcriptional elongation is shown here. Which is that promoter proximal pausing is extremely prevalent in melanoma, and not just in melanoma, but now we recognize in many cell types. So this is a ChIP-seq plot where we're looking at the amount of bound POL2, in this case across the snail gene, and as I said here, the traveling ratio is quite high, it's 18, and that's because the gene very effectively initiates, but really minimally elongates, so there's very minimal elongation, and so this would be caused, essentially classified as a pause gene. Whereas if you look at a gene like SOX10, an important neural cross transcription factor, there the traveling ratio is 3. There's tons of POL2 in the promoter of the gene, but also tons of POL2 in the body of the gene. And so I think that this is becoming a very, or has been for a number of years, a very obvious mechanism by which cells adopt different fates, because they are really poised for transcription by binding lots of POL2 in the promoter and then pausing right past the promoter site, and really called promoter proximal pausing. And what does leflunomide do? Well, this is one example gene of what it does, and this is the gene NPM1, and you can see, and this is our control cells shown here. This is the promoter peak of POL2, and you can see lots of POL2 throughout the body of the gene. This is an actively elongating gene, and in the presence of leflunomide, again, the POL2 promoter peak is identical, but what really changes is the amount of POL2 throughout the body of the gene. And so this is a really good example where the traveling ratio obviously increases, where it essentially affects proximal promoter pausing. It doesn't allow as much POL2 to escape the promoter region, and therefore leads to essentially transcriptional pausing at this particular gene. And when we look genome-wide, it turned out that leflunomide really only affects about 1,200 genes genome-wide, and this is what's usually called a Metagene Plot, looking across the entire gene body. Black is DMSO, leflunomide is in red, and what you can see is that our drug really does not affect, again, promoter initiation, but it really selectively affects proximal promoter pausing. It depletes POL2 throughout the body of the gene without affecting initiation at about 1,200 genes or so. What genes are they? I don't have time to go into a lot of the details. I'll just mention that there are really two classes of genes that are very obviously affected by our drug. First are the genes bound by the MYC transcription factor, and MYC is now known to have a very important role specifically in transcription elongation. The other important class of genes are those bound by MITF itself, and so taken together, both MITF and MYC-bound genes are about 30% of the leflunomide targets. So the mechanism by which the drug only affects these, really selectively affects this class of genes, is something we're obviously still investigating. We don't know the answer quite yet. The last question, I'll just finish off with the last couple of slides, is what does this do to the phenotype of melanoma cells? And sort of as predicated by the basis of our screen, we expected that the drug would be selective, although not completely specific for melanoma cells. And so these are 3 different melanoma cells in the presence of our drug, and these are just proliferation assays. And you can see a very nice dose-response curve to leflunomide over these cells, where when we look at other non-melanoma cell lines, these are 2 pancreas lines and a breast cancer line, we see very little, if any, effect on proliferation from the drug leflunomide. Interestingly, we had reasons to think that in melanoma there would be synergy between inhibiting BRAF and inhibiting DHODH with leflunomide, and that's because the enzyme directly upstream of DHODH is the enzyme CAD, and CAD is known to be directly activated and phosphorylated by the MAP kinase pathway, which would obviously be upregulated due to the BRAF mutation. And so we predicted that putting a BRAF inhibitor along with our drug would have additive or perhaps synergistic effects on affecting melanoma phenotypes. And so we tested this using nude mice xenografts. And so this is our control graph shown here, and obviously the tumors get bigger. In the presence of very low doses of the BRAF inhibitor, PLX 4720 in this case, we see moderate decrease in tumor growth. Our drug itself, leflunomide, again at a moderate dose, also causes a diminution in tumor growth. And when we put the 2 together, we see actually quite a great deal of synergy between inhibiting BRAF and inhibiting DHODH with leflunomide. And this is just a representative mouse shown here. And so we are currently interested in pursuing clinical trials of this since leflunomide is approved for rheumatoid arthritis, reasonably well tolerated. We think our preclinical observation from the zebrafish and mouse work will lead us to a phase 1/2 trial in combination with the BRAF inhibitor. This is really the way the trial is being designed. So this is being clinically led by Keith Flaherty here at MGH with Steve Hody at Dana-Farber. Really, the primary endpoint is an increase of progression-free survival of 50%. What's really interesting from my perspective, or our perspective on the trial, is that we've incorporated in ChIP-seq on the biopsies, because we think our drug primarily works by affecting elongation at those 1,200 loci. We really want to know, are the patients who respond those who show elongation defects at those 1,200 loci? And so all patients will be enrolled, confirm BRAF. They'll get a pre-treatment biopsy, they'll get leflunomide for 2 weeks, get another left biopsy, and then they'll get combination and get the 2 together and get a 3rd biopsy. And essentially this will let us do ChIP-seq for RNA pol2 and calculation of the traveling ratios, as I showed, to really ask, do people who respond, are they the ones who show the most robust defects in transcriptional elongation at those 1,200 loci? And so this trial hopefully should be opening reasonably soon. And so I'll just finish by saying, the zebrafish is a relatively newcomer in the field of cancer biology. I've shown 2 example genetic and chemical screens that gave us 2 interesting targets, ZTB1 and DHODH. And we think that obviously, hopefully we'll get more creative about the different types of screens. We do think it's a pretty good platform or ideal platform for high throughput oncogenomics. And obviously, we're excited to see that something that came out of a zebrafish screen could enter a clinical trial, at least in melanoma in the near future. I'll just finish by thanking a couple of people at Children's Hospital. Obviously, Len Zahn, whose lab I postdoced in, has really pioneered the field. Yuriv Hooveras and Craig Seal, who really did the entirety of the SETDB1 work. Our collaborators at the Whitehead, including Rick Young at University of Michigan and Sean Morrison's lab. And I'll be happy to take any questions.
So, quick question. So, if you're affecting elongation, how do you, your data would suggest 1,200 genes. Doesn't that seem a little surprising if you're hitting elongation globally? Like, yeah. How do you explain that?
Yeah, I think probably the most likely explanation is that the enzyme which we're targeting is probably only expressed in a subset of certain cell types. And I think we don't know yet, and I don't have the answer to this, whether the DHODH enzyme itself is in a complex with other elongation factors, and that's one of the things we're looking at. We just don't know the answer yet.
Okay.
So.
questions. Richard, how does leflunomide work in rheumatoid arthritis?
I mean, the presumption is that, so DHODH was originally described as an enzyme in the biosynthetic pathway for uridine, and T cells are known to have high need for uridine production. We've looked and saw if uridine explains most of the phenotypes, but it doesn't.
So it's like methotrexate, which works in rheumatoid as an anti-metabolite, maybe.
Yeah, so, but when we supplement uridine or UMP in our melanoma cells or in our neural crest cells, it doesn't reverse.
It's a totally different mechanism.
So we think it's a different mechanism than that, yeah.
I've heard that there's some interesting genetics about SETDB1. Do you want to talk about that, or is that published yet?
I think it's not published yet, so
okay.
I think Len would be happy to talk about it, but probably not yet.
Right. So, okay, everybody look for it when it comes out. So. Other questions? Yes.
It's a great talk. So for the second part of your talk, you link to inhibit to the transcription elongation phenocopies SPT5 phenotype. So it's not very clear for me how DHODH enzymatic function can link to the transcriptional elongation.
Yeah, as I said, that's one of the things we're interested in looking at right now is whether DHODH, because it appears to have this function outside of uridine synthesis, is involved in a complex with that. And so we're looking at that right now to see where in the complex it's likely to be. And it very well may be in a complex with SPT5 itself. And that's sort of the pathway we're looking at right now.
Just two quick questions on the SETDB1. So for the senescence, is it specific to BRAF, or it does impact on replicated senescence or the RAS-induced senescence? And the other thing I think I missed, we probably already talked, in the melanoma case, I think you mentioned it's high in the Levi. So does SETDB has to be higher than certain level in order to kind of get over the Levi?
Yeah, that's a good question. So I'll address the first one. We think that it's essentially somewhat independent of BRAF, and because there's no essentially or very little correlation between having a BRAF mutation and having a SETDB1 amplification. So those are not strictly linked to each other. So it's probably an independent effect. The second question, I'm blanking. Just repeat the second question really quickly. It was
level in the Levi.
Oh, right.
Yeah.
So the Levi generally have intermediate levels, and so yeah, the thought is that you need a certain amount of SETDB1, at least in tumors that amplified, to overcome the senescence due to either BRAF or potentially from NRAS as well. Although we haven't spent a lot of time looking at NRAS lesions. So.
Is there any indication from clinical samples that SETDB1 is mutated or amplified, or anything happened to it in human melanoma?
So the original screen was based on a SETDB1 amplification. So we know that in 20% or so it's amplified. The question of mutation has been looked at, and as far as I know, and I could be wrong about this, last time I looked there were no mutations in SETDB1.